Hi, in this video we'll resume our discussion of transcription, and in the previous video we had just concluded talking about transcription start and stop sites, which we now talk about in more detail. These are blowups of those regions. Here we have the transcription start site. That's where we're going to start making RNA. I want to mention that it's not necessarily, and in fact usually isn't, the translation start site, meaning we're not going to start making protein right here. We're going to start making protein somewhere down here. But we're going to start making RNA here at the +1 site. This region upstream of where the gene starts is called a promoter region. It is also called an operator region, and some people make distinctions between them, operator being sort of closer to the start site, maybe the first 100 or couple hundred base pairs, whereas a promoter can be thousands of base pairs upstream of the transcription start site. But an extremely important part of any promoter operator is the 35 bases immediately upstream of the start site, and this is called the TATA box. And the reason it's given that name is because of the preponderance of T's and A's. The area highlighted in green is conserved, but the high preponderance of T's and A's in this region. Notice that the TATA box has a characteristic spacing. It's 10 bases upstream of the transcription start site, and the other part of it is at -30, starts at -35 base pairs. Now, you know that T and A bases will unzip a little more easily than G and C base pairs. I should also add, you know that it's not because there are 3 hydrogen bonds in GC and 2 hydrogen bonds in T and A, but rather the stacking interactions. But they do unwind more easily, and so it's thought that one of the reasons you have lots of T's and A's in this region is to help this region open up more easily so that the polymerase can start making the mRNA molecule. The termination or stop signal is less well characterized, but it has this characteristic runs of T's right before the stop, and frequently there is a hairpin formation which will help the polymerase, signal the polymerase to stop transcription. But I'd like to say a little bit more about the TATA box. Here you see it in, this is a crystal structure of a protein which helps initiate transcription in blue and green. This is called the TATA binding protein. And when this crystal structure was solved, people were rather amazed at the very significant angle of about 60 degrees, as I recall, at which DNA is bent. This bending ability at this sequence is a consequence of the AT-rich nature of this portion of the nucleic acid, and highlights the fact that other characteristics rather than just the hydrogen bonding possibilities in the major and minor grooves contributes to sequence-specific DNA recognition by proteins. In this case, it is the bend of the overall structure. This slide shows that genes can be coded on either strand of the genome, and that they are oriented in the same direction on that strand, because genes are always coded for such that the mRNA will be synthesized in the 5' to 3' direction. Also notice that the genes can overlap when they're on different strands, but that is a rather rare event, meaning that there would be, if this gene were extended a little further, there would be a region of overlap between gene D and gene C. This slide highlights the fact that the RNA polymerase is not a single protein, but rather a complex machine with lots of different protein parts. You won't be responsible for knowing most of these, but it's important to see. In general, all these proteins, TF2D, TF2B, E, and H, these are known as transcription accessory factors, or TAFs, and they are utilized at all all transcription start sites. The initial binding event to start transcription is the TATA binding protein, part of the TF2D complex, binds to the TATA box. This helps recruit other factors like TF2B, the RNA polymerase complex, and other factors, and now this complex gets assembled at the transcription start site and transcription can begin. The book sometimes refers to these as transcription factors. Most biologists would not call them transcription factors. That's a term reserved more for proteins that bind only at a select few number of genes to turn them on specifically. The last point to make is that these complexes, these proteins assemble at the transcription start site, but as transcription starts, most of them will be removed from the complex and the polymerase will go on by itself. The gene-specific factors, or real transcription factors, occur at regions called enhancers. These can be very far upstream from the TATA box. In fact, many thousands of base pairs away. And for a long time it was wondered about how a protein binding thousands of base pairs away could influence proteins binding here at the TATA box. It's almost embarrassing in retrospect, but simply by the DNA looping around, these proteins can be in immediate contact with the rest of these proteins and all contribute thermodynamically to a greater binding energy to assemble a complex at a specific location to turn this gene on and for transcription to occur. On this slide, we're going to highlight some of the differences between prokaryotes and eukaryotic mRNA. There are at least three important ones here. We'll start with the eukaryotic mRNA. First, there is a cap called a 7-methyl G cap. The 7 is the location on the G to which the methyl is attached. This cap structure is important for proteins to bind there and for stability of the sequence. Also, every transcript ends with called a poly A tail of approximately 200 bases. Eukaryotic mRNAs almost exclusively code for a single protein. And that's the biggest difference for prokaryotic mRNAs. These are sometimes referred to as operons, and a single mRNA transcript can code for 3 proteins. There will be 3 different translation start sites. Ribosomes will bind here, here, and here, and they'll make multiple proteins from a single mRNA transcript. These proteins are almost always functionally related. Like in this example, it might be 3 proteins all involved for a particular type of amino acid synthesis. This is often referred to as mRNAs in bacteria are polycistronic. This slide highlights another important difference between bacteria and eukaryotes, and that is that bacterial genes almost exclusively only have coding regions. Every RNA, here's the transcription start site, here's the translation start site, and this entire RNA stretch codes for protein. In eukaryotes, there are different regions called exons and introns. The exons code for protein, the introns are intervening material, and they have some functions, but less is known about them. The discovery of introns was performed by Richard Roberts and Philip Sharp. Dr. Roberts is an adjunct professor here at Boston University, and he received a Nobel Prize for his part in this discovery. So in eukaryotes, what will happen is the organism will transcribe the entire gene. It will then cut out the intron regions, and you'll be left with a final transcript that is much shorter, but then this entire transcript here, a mature mRNA, will be code for protein. The size of human eukaryotic proteins can vary quite a bit. Some can extend over extremely large regions, in this case 200,000 base pairs, and you can see that most of the sequence is in fact intron. And there are examples that are even larger than this that extend over 1,000,000 base pairs. One of the potential advantages of using many exons to code for the protein is you can incorporate some modularity, and these transcripts can be alternatively spliced. That means in different situations you can utilize different exons. Even though all of them are initially transcribed, you can combine them in different combinations to make more different types of protein from a single RNA transcript. There are many different kinds of logic that can be incorporated here. Just about anything you can think of can be used. In this example, exons 2 and 3 are used in an either-or fashion. Some of the final exons of multiple, each is just you pick one for a different 3' end to the gene, and sometimes exons are skipped altogether. So that's what I wanted to say about transcription. We'll move on to translation with the next video.